Hello, AP Bio folks. Today we are going to take the data that you collected yesterday and work through a lab together, looking at these properties of water and whether soapy water and rubbing alcohol make a difference in terms of cohesion on a penny. And we're also going to learn a little bit of statistics as we do this. So take a minute if you need to and go back and review the information yesterday about basic chemistry and some of the properties of water that are involved in this water sticking together on a penny. Now I want you to take a look at a couple of chemical structures. This is also some review from yesterday. You can see we have water here. The one in the upper right is isopropyl alcohol or rubbing alcohol. And the one at the bottom is one of the active ingredients in dish soap. As you look at these, I want you to think about whether these molecules have any polarity and whether that will affect their ability to cohere with water on a penny. And you're going to use this information to make a prediction. As we go through this lab today, I'd like you to take out a notebook or some graph paper and actually write this out. This lab is kind of part of your notes on statistics because it's kind of your practice run through how to do some of these calculations. So grab your notebook right now and write down your prediction. Which of these three liquids do you think had the most drops of water on a penny and the median amount and the least, and explain why using a little bit of science? So here's the data that you all collected for me yesterday. I took all of the samples for each of these trials and averaged them. So this is the average number of drops of water on your paper. And if you're just doing this in a notebook, that's okay for this one because you're not turning this in. Or you can grab a sheet of graph paper, make a quick graph of the data that you see here. You should have ended up with a graph that looks something like this. For this type of data, a bar graph makes the most sense because it's three completely separate trials. You can ignore the label on the x-axis. I just noticed that says tap water, and that doesn't make much sense. Based on this graph, now I want you to take just a minute and jot down a quick conclusion. Looking back at your hypothesis, were you correct? And which of these does in fact have the most cohesion and the least and all that kind of stuff. So write a quick conclusion to this. So your conclusion here was probably something to the effect of tap water shows the most cohesion and had the highest number of drops, followed by alcohol, and soapy water had the least. But the important thing today is that as we get further into science, up to this point in science, probably what we just did is how you have interpreted data. You found an average and made a graph and used that to base your conclusion on. But averages aren't always enough. Take a minute and read through this fun little story and see if you get the joke. When we're trying to figure out whether data is really significant and whether differences are big enough to really matter, we need to do a little more work than just calculating an average. And that's what we're going to talk about today. Descriptive statistics is what we call these types of calculations to give us a clearer picture of what this data really looks like. We're going to look at a few different ones. You may have heard of standard deviation. Standard deviation just describes how much variability there is in the data. So when you look at all of your data, are your data points really spread out? That would have a high standard deviation. Or are they all fairly close together within a small range of numbers? That would have a smaller standard deviation. You've heard of other measurements such as the mean, the median, and the mode. The one that's probably brand new to you today is standard error of the mean. That's the one that we're going to spend the most time on today. As we go through and learn about these, The important things to remember is that you need to understand what these mean conceptually. You need to understand that standard deviation is a measurement of variability, how spread out the data is, and we're going to talk about standard error here in a minute. We are going to calculate these a few times for some labs in class, but you will not be asked to calculate them on a test, and you will not be asked to calculate these on the AP exam. You might see questions about these, but you won't be asked to calculate them. The things you will need to do is you'll need to be able to understand what these values tell you about the data. You'll need to be able to make graphs that include standard error bars, and you need to be able to interpret those graphs. So those are the skills that we're going to focus on today. As we go through this lab, I am going to do the calculations on a Google Sheet, and I'm not going to spend a lot of time explaining how to do these calculations today, because there are a few different ways that you can approach this. But if you want me to show you either how to do it on a spreadsheet like this or on a calculator, you can ask me later. So standard error of the mean helps us to figure out how reliable our data is. And so if you have data that looks like this, the top graph here shows some data about caffeine concentration affecting change in pulse rate. And you can see there are several data points for each concentration. This bottom graph is what it looks like if you average those. You get this pretty clear pattern. It goes up and then it comes back down. But when we look at standard error, it basically shows us the reliability. And you can see the error bars that are on here give you an idea of how reliable that data is. The one over here on the left is a fairly small error bar. That tells us this data is pretty reliable. It has a fairly small range. It's all pretty close together, as opposed to this one at 10, which is very spread out. It's got this big outlier up here, which makes us doubt the reliability of that. It's much more spread out. And you can see anywhere that the data points are further spread out, we end up with a longer error bar. You can also see this last one, the last data point at 25 millimoles has a really big error bar. That data point is less reliable because there are only 2 points there. And so if we have less data, we can't trust it quite as much. So that's kind of what standard error tells us, is how reliable our data is. When we use standard error in biology, we always calculate plus or minus 2 standard error of the mean, which you'll either see written like this. This X should actually have a bar over the top of it. Let's see if I can draw that in here for you. It should have that little flat line over the top. That means average. Sorry, that doesn't look real great, but that's what that's supposed to be. You will also see it written as capital SEM. So if you've seen curves like this before, this is a normal distribution. And if you go plus or minus 1 standard deviation or standard error away, they're not exactly the same thing, but the distribution in a normal curve is the same. That includes about 68% of the data underneath this curve. Pretty good, but we would like to be a little more than 68% confident when we talk about things in biology. If you go 2 out from the mean, now you cover 95% of the data. We could even go 3, which would make us pretty close to 100% confident in this data, but we don't need to be quite that specific. Usually in biology, we use plus or minus 2 standard error of the mean, which gives us 95% confidence that our data is statistically significant. And so in biology, you will always draw error bars at plus or minus 2 standard error of the mean, unless the directions specifically tell you to do something different. When you draw these error bars in here, I'm going to wait a second for that thing at the bottom to disappear so that you can see this. When you draw error bars, like I said, drawing the error bars is a skill that you need to have. You will usually be given a data table like you see at the bottom of the screen right now, where it gives you the standard deviation and it gives you the standard error. what you will need to be able to do is need to be able to look at that data and notice, first of all, this is one standard error of the mean. And like we just talked about in biology, we always go up and down 2 standard error of the mean. So you need to double that. So for this first column, 2 times the standard error would be roughly 0.26. And so what that means is we're going to take our average, which here is about 0.735, and we are going to go up from there 0.26. Let's see if my brain can still do math. So 0.73 plus 0.26 would be about 0.99. Sorry, it's getting late. So my error bar is going to start here at the average, and it's going to go up. My graph doesn't even go quite far enough, up to 0.99. We'll say it's about there. But it's also going to go down to standard error. So we're also going to do 0.73 minus 0.26. That's going to be 0.47. So my error bar is going to go down to about 0.47. That's about there. So this error bar is going to go up and down. It probably should have gone up a little bit farther, but my words got in the way there. So that's what our error bar would look like there. We can do one more just for practice. If we double this, 2 standard error for the second error, or the second data point would be about 0.14. So we are going to take our average, which is right here, and we're going to add 0.14. So that's almost 0.38 plus 0.14, be 0.48, 0.52. So our error bar is going to go up to about 0.52 right there. Sorry, I can't draw real straight lines. And it's also going to go down by 0.14. So 0.37 or 0.38 minus 0.14 is going to be about 0.24, right about there. So our error bar goes up 2 times the standard error and down 2 times the standard error. Now, this is probably the most important part, is analyzing these error bars. And what you want to look for when you look at a graph is you want to look at whether or not the error bars overlap. What these error bars really tell us, the easiest way to think about this, when we did this experiment, whatever this particular graph is showing you, we calculated the average right here. But as you know, if you do an experiment again, if you guys went back and dropped water on a penny again today, you probably wouldn't get exactly the same average that you got yesterday, right? And so what this error bar really is telling us is that even though we calculated this average, based on the data that we have, the true actual average, if we could do this experiment a million billion times, the true average would fall somewhere between here and here. And this one would fall somewhere between here and here. So it's possible, if you look at that, hopefully you can see that it's possible that the average of this first one, of population 1, could be right here. And the average of population 2 could be right here, which means they could actually be completely the same. So what you're looking for here is whether the error bars overlap. That's the most important thing to pay attention to. If they do not overlap at all, for example, population 1, the error bar goes to here. Population 3, it doesn't start to here. So you can see, even if we trace those all the way over, I'm not going to draw a real straight line, sorry. Whoa, that went off. If we trace that all the way over here, those two don't overlap at all. So if they don't overlap at all, we say we can be 95% confident that there is a significant difference between population 1 and population 3. There's some important language in this, and whenever you write conclusions that have to do with error bars, you need to include that you are 95% confident, and you should also include The reason you're 95% confident is because the error bars do not overlap. So you can see down here, it kind of gives you the language for how you'd write this. The difference between population 1 and population 3 is statistically significant. Or you can say, I am 95% confident that population 1 and 3 are different because the error bars do not overlap. So like I said, these are kind of the two key phrases to put in your brain when you're writing about error bars. If the error bars do overlap, we can be pretty confident that there's no real difference. Like I said, population 1 and 2, the actual average of these could be the same because they overlap quite a bit. And the same with populations 2 and 3. There may not be any real difference there. The only ones that we can be confident are different in this particular case are population 1 and 3. So if we come back to look at this graph that we started with, you can see a couple more examples of that. If we look at 20 and 25 millimoles of caffeine, there's so much overlap between these that they may not be statistically significant. This error bar goes down to here, this one comes up to here. So you've got this whole area of overlap where the average could actually be the same. And this is in the wrong spot. I think that was supposed to be looking at these two. Sorry, I'm not sure what happened to that slide. I think that was supposed to be looking at these two error bars. They have very little overlap. So it is much more likely that those two are significantly different because the error bars do not overlap. So that's a little bit about how to draw and interpret error bars. We're going to go back and add those into our Penny lab now and see if that data really is significantly different or whether those differences are just due to kind of random chance and random differences. So I'm going to pull up the spreadsheet here. In this spreadsheet, this is where I went through and I entered in all of the data you collected. So we have our tap water, soapy water, and rubbing alcohol. And I just entered all your data. You can see alcohol has the least data. Only a few people did that one, but thank you to the 4 or 5 of you that did. We have more data on soapy water and lots on tap water because everybody did that. These are the averages that I already calculated and that I used to make that graph up there. So the next thing that I'm going to do, the equations for calculating these, first of all, are found on the equations and formula sheet. If you haven't looked at this yet, this document is found under the class materials in Google Classroom. And this is something that you will want to refer to pretty frequently throughout this class. They have these stars next to them because, like I said, you will not need to calculate these on the AP exam. I would strongly recommend that if you have access to a computer with a spreadsheet or a graphing calculator, that you never try and do this calculation by hand ever for standard deviation. It's possible. It's just really tedious and slow. This one is not so bad. So if you can get a computer to do the standard deviation for you, the standard error is equal to S divided by the square root of N. And it's important to notice over here that on the right-hand side of this equation and formula sheet, it tells you what these variables mean. S is the standard deviation of your sample, which, like I said, you can calculate on a computer or calculator. And N is your sample size. So standard error is determined kind of by those two things. How variable is your data? What's the standard deviation? And how big is your sample size? Because the bigger the sample size, the more we can trust that data. So we are going to use this calculation and we're going to go back over to our data and try and figure this out. So first I'm going to calculate the standard deviation of each of these data sets. And I'm going to do this on a spreadsheet. There's a video that I will make available to you later that shows you how to do this calculation, depending how familiar you are with Google Sheets or Excel. It is actually really easy to calculate things like this. So this is our standard deviation down in the bottom left. For the tap water, it's about 7.1106. I'm going to calculate the other two really quick. And you might be able to see what I'm doing. You just have to type equals STDEV is the standard deviation code. And then you put parentheses and highlight the boxes for which you want to calculate the standard deviation. For this one, I got to scroll up a little bit. Here we go. And it will do this calculation for us. So there's our standard deviations, and I'm just going to get rid of a couple of those extra decimal points so it doesn't look quite so cumbersome. And so to calculate standard error, so standard error of the mean, SEM, that's what that stands for. The equation, as you saw here, was the standard deviation, S, divided by the square root of N. That's the sample size. So our sample size for this first one is 70. You can see there's 71 rows of data for tap water, but the top one is just the label. So I'm going to grab my calculator. Oh, look, I think I just did this actually. So square root of 70 is about 8.37. So I'm going to go like this. So standard error of the mean equals our standard deviation divided by the square root of 70, which is about 8.37. So there is our standard error for that one. This one has 65 data points. So square root of 65 is 8.06 divided by 8.06. And the last one, how many data points do we have there? 20. Square root of 20 is 4.47. So all right, so this row now is our standard error. I'm also going to use this to figure out 2 times the standard error. I'm just going to do all my calculations in here. Just because that's a little bit faster. So this is the number that's going to be most important to us in our graph. You can also, sometimes it's helpful to kind of make yourself a table, because really when we graph this, we already have our bar graph made with the averages with these 3 numbers. And we're going to want to include our error bars that go up to standard error from that number and down. So we can calculate that as well. So this row is going to be the average plus 2 standard error. And then I'll make a row that's the average minus 2 standard error. And that will just make it really, really easy for us to graph. So this will be equal to the average plus 2 standard error. I think I can just copy that calculation over. Awesome. And this one will be equal to our average minus our 2 standard error. There. All right, now I'm going to see if I can drag our graph down here so that we can see it. I'll make that a little smaller. See if I can get this all on one screen for you. Here we go. So now our error bar, what we've calculated here, our error bar has to go from the average, which is where our bar is, up to standard error of the mean. So for this first, for tap water, it has to go up to 30.627 and down to standard error. So it has to go down to 27.203. So I'm going to draw that error bar in here. We want to go up to about 30.627, which is going to be right about here. And down to 27.23. And I don't have a lot of lines on this, so I'm just going to estimate right about there. If you have notebook paper or graphing paper or something, hopefully you can do this a little more precisely. It is important to be fairly precise with these so that you can see if the error bars overlap. On the soapy water, our error bar is going to go from 20.291 right here, right about there, down to 17.586. And on isopropyl alcohol, our error bar is going to go up to 29 and down to about 25. We have a little larger error bar on this one, mainly because we had less data. Remember, not as many people did alcohol, so our data is a little bit less reliable. So now when we take a look at this, you can see that we need to refine our conclusion a little bit. Take a minute and rewrite your conclusion. Look at whether these error bars overlap, and talk about whether this data actually is statistically significant or not. Hopefully you noticed as you looked at this that if you look at the tap water and the alcohol actually is not significantly different from the tap water because those error bars overlap. You can see that in the numbers here. The bottom of the tap water is at 27. The top of the alcohol is at 29. So those definitely overlap each other. So your conclusion would say something like this. Alcohol is not significant. Hang on, I can't talk and type at the same time. Significantly alcohol is not significantly different than tap water since the error bars overlap. Hang on, I'm going to reword that. The difference between soapy water and tap water and between soapy water and alcohol is statistically significant at 95% confidence because the error bars do not overlap. If yours isn't in exactly those words, that's okay. But remember, you should have that 95% confidence in there somewhere. And you have to talk about whether or not the error bars overlap. Every single AP question that I have ever seen about error bars, you get the point if you talk about whether or not the error bars overlap, and you don't get the point if you don't talk about that. So that's the most important piece when you're interpreting graphs that include error bars. So just to wrap up today, keep what you wrote about that penny lab. This is not a lab we're going to turn in. This really was kind of a chance to practice these statistics because we're going to do the same type of interpretation on another lab next week and use standard deviation and standard error again. If you want some more review on these, there are 2 videos linked on today's Google Classroom post and a Google Doc that you can use as a note sheet. You can make your own copy of it and type right in there if you would like to go through and review these statistics if you feel like you need a little more help understanding exactly what they mean and how to calculate them. You can also see on the screen here, there are some reminders of some of the things that are coming up. Next week, we are going to be moving into kind of our last topic. We're going to pull this all back together and talk about how the chemistry of water helps it to move through plants. And so you can start looking over that next section. I hope you have a wonderful weekend, and we will see you next week.